Okay, good morning, good evening, good afternoon, wherever you're joining us today. Welcome to our ISAAA webinar on genome editing, the series that has been running over the last 1 year. We are indeed very excited that today you are able to join us, and we are going to continue with the platform as we have been doing. And just to start us off, I want to thank everyone for getting the time to be with us. We are joined by a very well seasoned expert team. One is Diana Horvath from the US, Chicago. She's the president and the co-founder of a charitable organization by name Two Blades, which is looking at how best to deliver the promise of new technologies to farmers and also delivering new tools to agricultural biotechnology space. Diane is very passionate about solutions to the challenges that are affecting our agricultural sector, and most importantly, she's working with tools that enable to confer protection against the major unmanned diseases of major crops, and of course the major staples to ensure that we have supply and equitable access to scientific progress for commercial and for the smallholder agriculture. We also have with us Dr. Jimmy Botella. Dr. Botella is joining us from Australia. He's a professor from University of Queensland, a seasoned teacher who has taught many students over the years. Most importantly, Jimmy established the Plant Genetic Engineering Laboratory at the University of Queensland when he joined. And with him, he has 11 international patents in the field of plant biotechnology. And he has also founded 2 biotech companies and a member of the expert scientific panel in Australia. In today's topic of new breeding tools, at ISAAA, we share knowledge on probiotechnology, and importantly, we are doing this so that we can move forward through a system where we have more of dialogue than debates. Over the last 20 years, we have witnessed a lot of debates about plant biotechnology. We feel that by starting early, just by the wisdom of the African tradition, which says that when you start early, you shorten the journey. And the shortening of this journey is just making sure that we are getting informed policies and informed choices on the new tools that are in the global space and that farmers can have a choice. We are happy that this seminar is hosted by our Southeast Asia Center. And with us, we have the director of the Southeast Asia Center, Dr. Rhodora Romero-Aldemita. Ola, you can say hello. And then we are also joined by the teams from Southeast Asia and also from the Afri Center. And we are broadcasting from Nairobi, Kenya. So without much ado, again, a very warm welcome. We are happy that we have these 2 experts who are going to share with us about innovative tools for crop breeding, so that as we get into this area where so many of us want to discuss, want to contribute to the discussion, but we have also learned that over 80% of those who are engaging in debates, not dialogue, do not have an idea of what tools we have. So we are looking forward to getting more on what there is in the global space and how best we can utilize these tools. So welcome to Diana. You have the screen, Diana.
Thank you so much, Margaret, and Ola, and all the ISAAA staff. It's great to be here today. I'm talking to you today about TALENs as a versatile tool in crop breeding, and I'm presenting this topic from my experience over the last 24 years working on applications of molecular biology for agriculture. Initially from a perspective in a venture capital fund where we were focusing on startup businesses that were exploiting agricultural biotechnologies, and then from the perspective of Two Blades where we focused on genetic solutions for crop diseases. I'd like to describe some of the unique features about TALENs, how they were discovered, how they were put to use, and how they differ from other editing tools. To start that, I need to give a little bit of background about Two Blades and how we came to be involved with TALENs. So Two Blades is a US-based nonprofit foundation started in 2004 to translate advanced knowledge from plant science into crop disease solutions to improve agriculture and food security worldwide. And we have a two-pronged business model to tackling crop diseases. We work with seed companies on the one hand to develop commercial seed varieties with durable resistance to benefit production at a large scale. And we partner with appropriate government, international agencies, and charitable foundations to benefit smallholders and agriculture in less developed countries. And we've built an ecosystem globally with our own research group shown here, embedded within the Sainsbury Laboratory in the UK, and through academic collaborators, partners in industry, and international agricultural development agencies like CSIRO in Australia, and members of the consultative group in international agricultural research like CIMMYT, SIP, and IITA, all with the idea of creating a new model for advancing science for broad benefit. And our work with TALENs exemplifies this mission. We first learned about TALENs because they come from a plant disease, a plant pathogen, particularly the one called Xanthomonas. And Xanthomonas causes bacterial spot, a serious disease of pepper and tomato, and disease on a number of other crops. Scientists at Martin Luther University in Halle, Germany, shown here in the group of Ulla Bonas, together with Sebastian Schornock and Thomas Lahey and Jens Bach. And I want to just have a shout out to Jens for sharing some slides with me today. Give him credit for those. You'll see those marked with his name. They were working on a disease resistance gene in pepper called BS3. And I first heard in depth about BS3 at the 2007 Molecular Plant Microbe Interaction meeting in Sorrento. And Thomas had the unfortunate time slot of the evening session. A lot of people had already gone off for a nice dinner in town, but I'd heard about that this talk was going to happen, so I made a point of staying late. And Thomas gave an excellent talk, really exciting. Afterwards, I went and I grabbed him and we sat and we talked for a couple of hours, and I really got to learn about BS3 and this system, which was so fascinating. And these scientists, in the course of studying BS3, identified that the trigger for resistance is the effector protein AVR-BS3. And effectors are part of the arsenal that pathogens have to establish infection. They're proteins that get injected inside the plant cell, and there they suppress host defenses and promote genes that enhance virulence. Typically, plant resistance involves immune proteins that are sentries on the cell surface or inside the plant cell, and when they detect the presence of a pathogen, they trigger the innate defense responses. In the case of BS3, there were several really unusual aspects of this interaction that they discovered. First of all, the AVR-BS3 protein is a gene activator, and not one that works in bacteria where it's made, but one that works in plants only. And yet plants themselves don't make this protein, only the bacteria. So the bacteria picked it up. The second interesting thing was that the resistance in pepper doesn't work by the usual protein receptors, but by a decoy gene that evolved with an AVR-BS3 binding site in the regulatory region that when AVR-BS3 binds, it activates the expression of this resistance gene and leads to localized cell death that blocks the infection. But the most amazing discovery about this system was the structure of the effector protein itself. AVR-BS3 is the prototype protein for a class of bacterial effectors called transcription activator-like effectors, so called because they activate transcription or gene expression. And they have a central domain structure that has a series of repeats, generally about 13 to 20 of these. And this repeat sequence is highly conserved except at two positions that were more variable. And it was the Halle team that worked out that these amino acids specify binding of a single repeat in the protein to a single base in the DNA. And the adjacent repeats bound in a linear one-to-one fashion with adjacent DNA bases. So if there were 17 repeats, that means they would have a recognition sequence of 17 bases. And this is quite long compared to other DNA binding proteins like restriction enzymes. And it allows these proteins to have an exquisite specificity. The reaction, the interaction was akin to a kind of code, a codex in a way, where you could understand the protein sequence and DNA connection. So that, for example, if you have a asparagine isoleucine pair in the repeat, it would have a strong preference for binding adenine. So all you had to do now with this knowledge, you could look at naturally existing TAL effectors, and you could interpret what their binding sequence is, what they were naturally going in to target within the plant. And this helped to understand a lot about disease, but it also meant you could now design proteins to bind to any target sequence you want. And this was a great improvement in terms of being able to target proteins and functions and make changes in how traits are expressed. So in particular, we're here to talk about TALENs, and TALENs are a fusion between the TAL effector binding domain and a nuclease that cuts DNA. And typically, FOC1 is the nuclease that's been most commonly used. When you have a nuclease targeted to a stretch of DNA, it makes cuts in the DNA, and double-strand cuts trigger the plant's cells' innate repair pathways to, in the absence of a repair template. Let me just turn on my laser pointer here. In the absence of a repair template, just caused a process of end joining, which can either drop out sequences or, with an error-prone repair, make just small changes. But you can also add a DNA repair template, which can allow you to get more change and repair over a bigger area, or to add genes entirely. And so this sequence diversity allows the creation of new alleles and new traits, and that's very powerful. Comparing tools for gene editing, TALENs were a major breakthrough after following in the path of meganucleases and zinc finger proteins, zinc finger nucleases, not only in terms of the specificity and the ease to engineer, but also in the time to generate the molecules and the cost. It really brought all of these things down quite dramatically. Not long after TALENs were discovered, CRISPR became more widely known and used, and this had even greater efficiencies in terms of the time to produce and the cost. And Jimmy's going to be telling us a lot about CRISPR shortly. But even with the new popularity with CRISPRs, one of the key features about TALENs that remains distinguishing is the IP landscape and licensing. And when this discovery was made, Two Blades worked together with the HALA team to help exploit this TAL code discovery. Two Blades brought our experience with patenting and licensing, and we undertook the patent filing and licensed the rights for uses in plants. And then together with the inventors, we sought a major partner to develop the reagent uses to sell TALENs themselves for research purposes, as well as the commercial rights for therapeutic and industrial applications. And we chose Life Technologies, now Thermo Fisher, because they could do both of these activities on a large scale. This was consistent with our hope for getting the technology out there and accessible to a lot of people. So for Two Blades, we pursued then the crop uses and trying to make the TALEN platform widely available. And we worked with Ken Mooney, a consultant in the ag biotech space. And we set about to create a general strategy of non-exclusive licenses that was tailored by the licensee. If it was a commercial entity, we would scale the fees depending on the scale of the size of the company. We had just simple payments that were upfront, no downstream payments in terms of royalties or milestones. We had defined the rights that they had in terms of the number of crops they wanted to apply it to, or the field of use. So it could be used for double-strand breaks, or transcription activation, or repression, or a number of others, with the option to add more as the user would like. We also had a different approach for nonprofits, which is to license for no upfront fees. And for example, we licensed on that basis to the International Rice Research Institute for their purposes. And in all cases, we had grant backs of improvements for our uses and applications in developing agriculture. And overall, it was our aim to make this technology broadly available and the industry standard. And this model was unusual for other gene editing platforms for zinc fingers and even CRISPR, and was well embraced by all. There is one other set of rights held by Calyxt, and they have limited access to the technology, so some of the adoption hasn't been as widespread as we would have liked, but for our part, we've pushed hard to make the technology accessible as per our mission. So why editing? Editing is a really important part of the crop improvement toolkit. We need all of the tools available from conventional breeding right up to all the new breeding tools in order to accelerate breeding and produce more options for important traits with all the important challenges to agriculture that are currently upon us. And as we move towards 2050 and growing populations that need to be fed, and also with climate change, we need to have more ability to address traits like yield, architecture, and resilience to biotic and abiotic stresses. So talents are a versatile tool for this purpose. I've mentioned already that they can be used as activators in plants, but they can also be modified by changing out and fusing other domains to the end of the protein to modulate transcription in animal cells or repress transcription, to modify the epigenome. They're able to distinguish between methylation of DNA and to be used in the editing sense, and also for other applications like recombination. Crops that have been modified by talents, and this is just a sample, but soybean, rice, potato, maize, and wheat for a variety of traits, but typically they tend to be either quality traits, such as oil quality and reduced fat, reduced acrylamide, aroma, and secondly, disease resistance has been another target, like bacterial blight resistance in rice and powdery mildew in wheat. Talents have also been used for targeting in other systems. They've been successfully used in people to address conditions, diseases such as leukemia, and also in animal agriculture, as well as industry. And they have been used to edit in organelles. They can do that because they don't have a nucleic acid component requirement and can get inside organelles. There are some limitations for gene editing, and these include the number of target genes that can be accessed. From the disease resistance perspective, what we look for targets are so-called susceptibility genes, genes that when they're present can be exploited by the pathogen. And so if you can interfere with that process, then you build in resistance. And the number of so-called susceptibility genes is somewhat limited. Also, when you have a target that you want to edit, not all of them can just be knocked out. They may have housekeeping functions. And so the goal is, instead of knocking them out, finding a new version, a new sequence, a new allele. And that's a lot of research that has to be done to find the right allele, because some alleles may have detrimental effects by interfering with housekeeping functions. And we've seen this in some of our own work, that you get plants that may be stunted or it may even be lethal. There's still a lot of work on the development of delivery systems, different ways for getting these editing tools inside of cells. There's an interest in some cases of trying to get away from nucleic acid, for example, in Europe. And so regulatory policies are still catching up and are not uniformly amenable to disease editing yet. For example, in the EU, they treat gene-edited products the same as transgenics, as GMOs. But a last limitation that especially relevant to disease resistance is the limitation of the gene pool when you're just working within the genome of your target plant. For us, an important feature of breeding for durable resistance is to be able to find multiple ways to prevent a pathogen from getting hold and joining together multiple resistance genes into a plant. And if that gene pool is limited within that plant, you want to look to its wild relatives and related species to try to bring in other disease resistance genes and combine them into your crop, and preferably by targeting it into a single locus, as that will stay together when you do additional breeding. So one example of that was published recently, 5 transgene cassette that confers broad resistance in wheat to wheat stem rust, which is a disease that's plagued mankind in wheat production and agriculture since the dawn of agriculture. And in this approach, what this team at CSIRO did was to stack multiple resistance genes, as shown here, to create this resistance. And I want to be clear and point out that this was not done by TALENs or gene editing tools, but through a recombination strategy. But in future, this could be done by gene editing tools as they become more efficient. And in this study, what Michele and his colleagues did was to take genes from rye and wild wheat and goat grass and combine them in the system to produce the gene stack. And in field trials, it was shown that this produced very effective resistance. This is the level of disease on the susceptible types. This is the level of disease in the gene stack lines is undetectable. And these were some of the individual lines. And you can see that not every gene was effective against these strains. SR45, which you can also see here, and SR55 did not provide the level of resistance that you would need to put out, but together as a cassette, it was very resistant overall. And this shows a very important thing, which that transgenic crops are also an essential tool in the toolbox, and we really need them to achieve our goals for food security. We know they're safe, they're very effective, and they can provide long-lasting resistance, and they can save lives by protecting crops against chronic and catastrophic diseases. So we really want to come back to that notion of trust that we heard about in the ISAAA opening, to make sure that all the tools in the toolbox can be used. So in summary then, TALENs are a versatile and precise gene editing tool. There are new publications out all the time that describe faster, low-cost assembly methods. In some applications, they outperform CRISPR, but they're both very effective technologies. And TALENs have some unique aspects that they can be capable of nucleic acid-free editing, they can edit in organelles, and they can distinguish between methylated DNA and unmethylated DNA. And the licenses, as I say, are very accessible from TwoBlades. We're happy to talk with anybody about any applications that they might be interested in accessing the technology. And lastly, to learn more about TALENs, you can look at the ISAAA Pocket K number 59. ISAAA has been a great partner working to help us raise visibility about TALENs as an important tool, and we really enjoyed working with the whole team to generate the Pocket K. We also have information on our website. There's a Scoop It page run by Sebastian Schornock on the literature for TALENs, and then there's more information at the Thermo Fisher and Calyx websites. And that's where I'm done. Thank you very much.
Thank you. Thank you so much, Diana, for taking us through the science, the applications, and products in the pipeline. Key messages being that the breeding toolkit is expanding. We are getting new tools, we are getting more precise tools. Accessibility is obviously more enhanced. The cost effectiveness is still looking much better. And so we are seeing a situation where different tools will be applied for different situations. And again, another message is that no one tool that can do everything. It is each tool with its comparative advantage. And then thank you also for the opportunity to even get to discuss about the licensing process. Thank you. So the next speaker is Jimmy. I had already introduced him, and we are also joined by our global coordinator, Dr. Maha. No, she was not there when I was doing the introductions. Dear participants, please continue posting your questions in the Q&A, and we also mentioned that there will be an evaluation survey at the end, and this will also inform us about the certificates, because a number of the participants have been asking us about certificates. So thank you. So Jimmy, So Jimmy is going to talk about CRISPR, and he's also going to talk about the science, the products in the pipeline, and also potential applications. Thank you.
Let me get started. What I was saying is that I really thank you very much for inviting me to come to do this webinar. I was telling you guys before we started that ISAAA does a fantastic job. I think that most scientists are in the molecular space are really grateful to the job that you guys are doing. Today I'm going to talk about CRISPR for food production, for crop breeding. Okay. Let me start. Okay, let me start explaining in very simple terms what CRISPR is. All right, let's say that you want to create a mutation in this specific site into a chromosome. So CRISPR uses two elements. In lay terms, is a molecular scissors, that is a nuclease, and a homing device that's going to take your nuclease to the right place. Those molecular scissors are a number of Cas proteins, but the most common one is Cas9. And the homing device is a very small molecule of RNA. And that is the simplicity of the system. That is what makes CRISPR so accessible to everyone. In my lab now, we can clone 6 to 8 different CRISPR constructs in one afternoon. So it's really easy. So what happens is, let's say that you want to create a mutation in this specific site of a chromosome. Of course, you know the sequence. So all you need to do is you design your homing device. You design the guide RNA. That guide RNA, it got different lengths. Normally it's around 80 bases, of which 20 are the ones that will recognize the site. And you put those 20 bases in there, and it's going to bring your nuclease to the right position. And that nuclease will actually make a double strand break. So your chromosome will be split, and you're going to have a break in your chromosome. Now, that is not uncommon at all. That happens a lot. And the cell have a number of machineries to fix those things. So that repair is normally normally dealt very well with the repair machinery. In those cases, you will have the DNA, it just looks exactly the same. So you cut, but it's repaired perfectly. But sometimes what happens is that the repair makes a mistake. And when it makes a mistake, it introduces a mutation. And that imperfect repair and that mutation is what we're looking for in the basic CRISPR procedure, in the basic CRISPR technology. Now, let me make it clear. CRISPR is only the ability to take a nuclease to the very specific site in a chromosome and cut that DNA, cut that chromosome. The rest is a total natural repair mechanism that every cell has. There are things that cannot be done with CRISPR or TALENs or any kind of genome editing, and for which we will need just normal GM technology. But there are some difference between CRISPR and GM. The first one is that CRISPR does not introduce any DNA from any other organisms. The second difference is that CRISPR will actually introduce modifications with really high precision in very specific parts of the genome. And the third one is that once you have created a mutation with CRISPR, it's absolutely impossible to know how was it created. Is that a natural mutation? Was it done chemically? Was it done with physical means? It's absolutely impossible. There is nothing there left to tell you this was done as a result of CRISPR genome editing. What are the potential applications of CRISPRs? The thing is, I started writing them, and then I decided I'm not going to make a list because it would be too much. You can pretty much do anything you want with CRISPR. That's the beauty of it. With the original CRISPR, with the basic CRISPR, there are a number of things you can do, but there are so many new things in CRISPR that are CRISPR-based technologies that essentially is opening the door to just use your imagination. And I look for some of the stuff that's been done with CRISPR. I'm going to mention just a couple. I mean, CRISPR is most of the work done in CRISPR have been done in rice and in Arabidopsis. All right. But I'm not going to start with rice because we will be here till tomorrow, all right? So in tomato, for example, people have done CRISPR that actually done delayed ripening in 4 different ways. They have increased the anthocyanin content in fruits. They have increased the gamma amino butyric acid content in fruits. They have reduced antinutrients like oxalic acid. They have created things like fruit with pink coloration. And also they have created fruit with more lycopene and beta carotene content. All that has already been done, right? That's not on the pipeline. That has already been published. It's there, it's done. Potato, you have potato with high amylopectin. You have potato, with no amylose. There are potatoes now with reduced browning. When you cut potatoes, when it's supposed to air, the browning happens pretty fast. Now there are potatoes that don't get browning. And there are reduced black spots in potatoes. Some people have actually done potatoes that are lacking steroidal glycoalkaloids. And of course, one of the things about potatoes is the problem with acrylamide, what is pretty toxic, all right? But some potatoes have been done with reduced sugars. That actually means reduced acrylamide in chips. These are only 2 crops and some examples of the things that have been done, and they're not exhaustive. It's a lot more than that, all right? I'm gonna give you a quick one where you can see the potential of CRISPR and the potential application. This is something that I did with, all the credit has to go to the group in Shanghai of Dr. Jiankan Zhu, Professor Zhu, from the Chinese Academy of Science. I was lucky enough that one of my sabbaticals, I spent it in his group, and I actually put a proposal to the Academy of Science, and I got one of these professorial scholarships to be there working in CRISPR with him. And this is one of the projects that we're running at the lab, and we finish up, and it's a beautiful one. This is about converting normal rice into sticky rice. Now, we applied CRISPR. We know the gene that we wanted to target. The gene is called the WOX gene. And by the way, you have those natural mutations. But what we wanted to do is we wanted to get 2 elite varieties and convert it from normal rice into sticky rice and make sure that nothing else changes. So these are some of the plants, and the 2 varieties that we were using. One is called 9522 and the other one is called XS134. They're very popular in the Shanghai area, in the provinces around Shanghai too. We targeted this gene, and these are some of the mutations that we obtained with CRISPR. These are typical mutation of CRISPR. In this case, CRISPR actually cuts right there. All right, so this was, in red, you can see the target sequence. This is what's called the PAM, the proto-spatial adjacent motif. It's just a technical thing. We got one line that contained an extra A in the gene, another line contained an extra C, one contained an extra T. This line was missing 1 nucleotide. This line was missing 7. This line was missing 47 nucleotides. These are the variety, similar kind of things. You got one with an extra T, an extra A. This one was a bit of a mix. It got a lot of mistakes were made in this one. But the result is that those mutations actually interrupt the gene. And that gene is an important one because it's the one that controls the levels of amylose and amylopectin in the rice seed. And that is what actually controls the quality of the rice once it's cooked. So we actually produced those varieties, and we actually took them to the field. These are the varieties, the normal one and the genome editing. And this is the other variety and the genome editing. And if you look at these pictures, you can actually see that there is almost no difference. You can't see any difference in there. When you get the seeds, you cannot differentiate the seeds. They're absolutely the same in width, same size on length. For the other variety, exactly the same. And then we took them to the field. And when we took them to the field, we got absolutely the same yield. And these were actually 2 years in a row, 2 crops in a row. So the yield was absolutely the same from the original and the CRISPR, the original and the CRISPR. Nothing changes. But there actually, one thing that changes, when you go to the total starch that you see in the seed, it's absolutely the same. One, two CRISPR varieties, that's the other variety, two CRISPR, nothing. But when you look at the amylose content, that's what the difference is. So you have the same amount of starch, but it's a different kind of starch. And that different kind of starch is what we're gonna make that rice be sticky rice. That changes the quality in cooking. Best part of it, that in the T1, in the T2 generation, we obtain a number of lines like this one, this one, this one, and these two that did not contain the Cas9 transgene into them, meaning the genes were out, but the mutation was still in, and the mutation is forever. The mutation is stable. So essentially, what we produced was we actually produced two elite varieties. We moved them from being normal rice to sticky rice, and nothing else changes. This is important, especially for small farmers. Small farmers grow their varieties. They know how to grow them. They don't want to change varieties, because if we as scientists come and giving a new variety, it might work in their field, it might not work. They don't want to run the risk, because their families depend on this. All right, this is if the experiment doesn't work for me, this is Jimmy. He's in Australia. He's still going to have a salary. But for a farmer, if your crop doesn't work, means that you run out of money, and you need to have money, for your kids' education, to buy food, etc., etc. In this case, we're able to give the farmers the absolute same variety, with one thing that is different, and that is the quality of the rice. And they now can decide whether, looking at the prices in the market, say, okay, this year I'm going to grow some sticky rice, but I don't need to learn how to grow this. I know when to water them. I look at them and say, oh, it needs a bit of fertilizer. That is fantastic, because otherwise we will need to give them a completely new variety, the sticky rice, and they need to learn how to grow it. Not only that, we're doing it in our here at UQ. Is something fantastic that you can actually do with CRISPR. Rice is a crop that's been studied for so long. All right, okay. But rice have a number of mutations that are related to yield. Okay, one mutation actually what it does is it produces a truncation in a gene, meaning instead of having the full protein, you have a smaller protein. That's one of the mutations that you can see in rice, okay? We actually found that in rice. Well, it was known in rice. So what we did is we replicated the exact same mutation in sorghum, because with CRISPR, it's easy to do it. And it happens, it works. Our results are showing, this is not even published yet, but our results are showing 10% yield increase. Why? Because we learn from rice and we produce the same mutation in sorghum. Other thing is, one gene at a time, that's a thing of the past. With CRISPR, this is an example, again, it's published with, I published that with Professor Su and his group. This is 6 genes at the same time. Now look at this. We analyzed 15 plants, and one of them have a mutation in all 6 genes. This is in a T1 generation. We're not talking about, doing 5 back crosses or anything. One T1 generation, 15 plants analyzed, and one of them had mutation in 6 different genes at the same time. Okay, that's the basic CRISPR. There are a number of things that are really exciting that are coming. One of the things that you can do now is base editing, meaning you change an A for a G. This is very specific. You change an A for a G or a C for a T. And this is very important because many of the traits that we have in agriculture are SNPs, are single nucleotide polymorphism. So it's not about knocking out a gene. It's about introducing one single change in one single base. And that can be done with base editing, not all of them, but most of them. Another thing that people are doing is gene replacement or gene correction, meaning changing a gene for another gene that actually works better. You can also control the expression of genes. That is done by targeting the 5' regions of genes where the promoter is. Now, this is really interesting, and it can be done with TALEN too, but with CRISPR, it's just so easy. Epigenome control. You control the level of methylation of the genome. And the funny thing is, I've seen results where people have actually increased the expression of a gene or decreased the expression of a gene by targeting the epigenome. Now, this is interesting because in this case, we're not changing a single base in the genome. We're only changing the methylation state of the genome. Meaning that if you sequence the entire genome, you're not going to find that there is nothing done with it. What actually means it's very difficult to police. RNA editing, it's also possible with CRISPR, not only DNA editing, but RNA editing. And prime editing, that this is the latest one, only a few months old, that essentially that allows you to introduce anything, anywhere. I'm going to tell you a little bit about prime editing. I'm not going to get too much in detail, but this is a really exciting development. What tying this prime editing uses a combination. Cas9 have been fused with a reverse transcriptase into one single protein, a very big protein. This is your RNA that is taking you to the right place, okay? It's taking this Cas9 to the right place in the genome. This is the guide sequence that will find the place in the genome. Well, this RNA has been now, is a lot longer, and it includes whatever you want to introduce in your genome. So what happened is, Cas9, if you look at the bottom, number one, Cas9 comes and cuts one of the strands. This Cas9 is what we call a nuclease. It doesn't cut both strands, only one strand. This strand is open, and it actually pairs with this end of the RNA that brought it into place. This end over here will pair with it, and this reverse transcriptase will copy your template. And once you copy your template, you will introduce exactly what you had in the template into your gene, into your DNA. This is the latest. It's not very efficient yet, but that will allow you to essentially do anything you want. Okay. So my conclusions to stay true to the 20 minutes is genome editing will allow us to produce better varieties, faster and cheaper than normal breeding. It will allow us to transfer and replicate traits from one crop into another. And I showed you the example of sorghum, meaning anything you learn in rice, you can transfer to sorghum, and you can transfer it to a number of grain crops. Or if you got anything in tomatoes, you can transfer it to potatoes. It's very easy now, anything that you learn in one crop, to reproduce it into another one. And what for me is very important is because it's so cheap and it's so fast, it actually allows us to help small farmers. Now, genetic engineering up until now, because it's so difficult and it's so costly to get all the permits and to commercialize a crop, as a result, what happened is you need to make a lot of money in order to have one GM crop, because it's extremely expensive not to produce it. It's not expensive to produce a GM crop. It's expensive to get all the approvals in order to commercialize it. But with genome editing, it's extremely cheap. Europe is not a very positive territory for this. What is interesting about Europe, because if you do chemical mutagenesis and you produce 20,000 mutations in any place in the genome, that's not a problem. But if you produce one mutation exactly in one place, that is a problem. There is no logic to it. And of course, it would allow us to respond to emergencies. I mean, think about COVID, where the same thing could happen. Well, the same thing is happening with bananas now, with the new Fusarium race. So if we have a way to incorporate something, genome editing would allow us to do it. Finally, if you want to know a lot more of that, this is a review that I just published like a month ago. It's free, so it's an open review. You're more than welcome to download the paper. This is the place where you can download it. It was a bit overwhelming because when I was invited, I said yes, and I should have never said yes. when I started looking at everything that has been done in CRISPR, it was overwhelming. You cannot imagine. At one point, I thought I was just going crazy. Ended up being more than 30 pages of a review, and I only put the most important bits in there. So if you want to know something in more detail, that's the review. And as I say, it's free access. And that's the end of my presentation. We'll stop sharing. There it is.
Thank you. Thank you so very much, Jimmy, for that stimulating presentation on CRISPR. Again, the possibilities are enormous. The rate at which the change is happening and newer genome editing tools are coming into the space is also quite amazing. And so we are looking forward to improved crop protection and also moving forward and getting these tools to farmers, given that the cost is also being reduced. So we have quite some engaging questions that are coming also in our chat. Please, participants, continue putting in the questions. And I want to hand you over now to our moderator, Dr. Rhodora, who is going to take us through the Q&A. Dr. Rhodora is our director for the Southeast Asia Center and also the Knowledge Center on Crop Biotechnology Director.
Thank you, Margaret. I'm very happy about the talks tonight because we have, through these two tools, we were able to highlight the uses of innovative tools in production, in food production, in yield, and now also in disease resistance in terms of talents, which Dr. Horvath told us. So there are a lot of questions actually, and one of the things that came out is that, well, in a summary of the questions that are in the Q&A, we can see that there are still some people who are thinking what's going to happen with genetic engineering now that genome editing is here. Would these tools be forgotten, or what could be the edge or the niche of the genome editing technologies now after genetic engineering? So can we please ask both of you, Diana first and then Jimmy?
It's a very interesting question, and it actually was why I made a point of referencing the fact that gene editing is wonderful and can do a lot of things that we can't do in other approaches. As Jimmy very nicely described, also, there are so many positive features about it, and yet it can't do everything, particularly the example that I gave for disease resistance. Really, we feel that the best way to make long-lasting resistance is to be able to bring in more resistance. So I do wonder if there is an impact on transgenic approaches given the popularity of editing. And I think it's important for scientists in particular to talk about the fact that all the tools have a role to play, and the safety aspects that we know about transgenics, and to find ways to get more rational regulatory processes that can reduce those costs in the regulation side, so that important tools of all kinds can be put to use for agriculture.
Yeah. Okay.
Yes,
One of the yes, Jimmy, please. Thank you.
There are things that can be done with GN that cannot be done with genome editing. That's clear. There is no question about that. And I don't think that GM is a thing of the past. As a matter of fact, GM is the future of agriculture. There is no way that, by the way, I mean, all the things about GM, you guys know that it's been already published, and all the sweet potatoes we are eating are genetically modified. And then some other team actually last year looked into more crops, and they found, I think it was 23 different species that have been genetically modified naturally by agrobacterium. So the future is GM.
Yeah. Okay. One of the things that I am very interested also is there's a question that can be answered by Diana, because we know that in nature we do not have insect resistance. and it can be done through genetic engineering, just like our BTs and BT corn and BT eggplant and all that. Is it possible to do it in genome editing?
Well, a lot of the insect resistance that we've had to date, and certainly there's many acres of BT crops out there, and that's been a very effective insect control approach. And there are all kinds of BT genes with different specificities. By and large, those genes aren't naturally occurring in our crops. So though we can use gene editing tools to introduce those as one strategy, those are technically transgenic. They're the introduction of a foreign gene. So gene editing tools can be very helpful for targeting foreign genes into a genome. And that's one way to use the approach more effectively. But it's unlikely that those are going to be considered gene edited in the near future. There's some places, of course, now that are adopting new policies that say if you bring in genes from plants that could be crossed by conventional breeding, but you use a gene editing tool, that will not require the usual regulatory process of a foreign gene. So things are shifting. We can hope that the tools will continue to improve and the regulations will keep up with them to allow these advances to have impact for agriculture.
Okay, yes, thank you so much. Yes, even in talents, that is also, in CRISPR, that's also possible, that we can introduce foreign genes using CRISPR and just targeting it to the right side of the chromosome. One of the things that I am very much amazed is when, Jimmy, you were saying that in just one target sequence, there were several knockouts, that you were able to target several parts of the genome so that you can get your desired trait. I forgot what trait that was. So how could it be possible? Is it the targeting sequence can target many different sites? Is that right? Is that
No,
correct?
what you actually do is you produce multiple targeting sequences. You can put in in one single cassette. We did put 6 different targeted sequences in one single cassette, and it worked very well, as you could see. In that case, we were targeting ABA receptors. Again, it was just a proof of concept that it could be done. But that's it. What you do is you use different. Now, what I'm doing now with crops that are polyploids, for example, is we're using one single targeting to knock out the same gene in all the different genomes. So you need to look for one specific sequence that are present in all the genes of a polyploid, and you can do that in one single go.
Okay, yes, I got it. We're still discussing the science, so hold on a minute. There's still a lot of questions. So we focus first on the science and then we proceed to the other stuff. One of the things that was highlighted in Diane's talk is that the delivery systems in talents. You're still maximizing the delivery systems and the reduction of types. What are the measures that you do so that you can get to those targets, the delivery systems and reduction of types?
Well, that is not unique to talents. So I think for any kind of gene editing tool, there's a lot of research to think about how to deliver them and how to make sure that they don't include a transgene. So if you can have transient delivery, especially of proteinaceous compounds into the cell, then that's a great step forward. If you can do that at good efficiency, then it's kind of a hit and run edit, and that doesn't mean that means you don't have to incorporate a gene. Of course, we're trying to avoid foreign sequences that will trigger regulatory efforts. So those tools and methodologies, I think, are advancing all the time. There's a lot of work, I think, for CRISPRs and talents to improve delivery methodologies. And then in terms of off targets, again, that's also just a nature of editing, that if you're trying to target a gene, in some cases you can knock it out and it'll only have the function that you want to change. But in some cases, there might be other functions of that gene. And so what you're looking for is really not a loss of a trait, but a gain of a trait by giving it a new feature. And that, again, can be very specific to the mutation. I can give one example. There's a gene in pepper called BS5. And BS5 is a recessive resistance. And the difference between the wild type and the recessive resistant gene is a 6 base pair deletion. When you have a deletion, the rest of the function of the protein is intact, but now it's no longer lending itself to susceptibility to bacterial infection, and you can gain resistance. But if you make broader changes, if you knock out the gene or you take out bigger chunks, then you start to see off-types. So you have to do some trial and error to identify which changes can you make without interfering with other functions. And that's just trial and error.
Yeah, but this is lesser trial and error than genetic engineering.
Yes,
Right?
in the case of CRISPR, in the case of CRISPR, actually, it's we are using virus for delivery to avoid to go through a full transgenic generation. CRISPR is a lot easier in the sense that it's easier to put it into a virus. We have a number of approaches. Now there are some new Cas. There was one that was discovered 4 months ago or something. What is a really small Cas? It's called Cas5. We just got it artificially synthesized. It arrived to my lab yesterday morning, and we're about to put it into a virus, because now we can actually put that into a virus with the guide RNA. And what you do is you infect the tissue with the virus, so you produce the guide RNA and the Cas protein, and then you recover from that tissue, you regenerate plants from that tissue. And that is the approach that we are pursuing with that. And in that case, actually, you avoid a lot of tissue culture. There are other people who have used viruses, but they're using tricks. Like the plants are Cas9 positive, so they only use the virus to deliver the guide RNA. But we are delivering the guide RNA and the Cas in a single virus. And we are hopeful that in a few months, we could actually have a method to produce Cas, mutations without, and there are RNA viruses, so they are not going to introduce any DNA into it. So there is a lot of development going on in there.
Yes, it's so exciting. Every day, if you go see our crop biotech update and our new breeding innovations, new breeding tools section, we always feature new news, and it's very fast how things come out. One of the questions on CRISPR is, can you explain further what is Cas9, Cas13D? This is
right. There are a number of Cases. There are Cas9, Cas12, Cas13.
Cas5, yes.
There are different types, essentially, all right? What is the difference? Some of them actually, Cas13 is an RNA editor. So Cas13 will actually target RNA. Essentially, from Cas to Cas, what is going to change is the sequence that they need to have. The PAM will be different from one Cas to another. And then the processing of the guide RNA. Some Cas enzymes can process their own RNA, what is really useful. Some other Cas cannot. Cas9, cannot process it, but Cas12, with Cas12, if you can put 4 different guides, one after the other, Cas12 will actually cut them into pieces and incorporate them.
Okay. So can you recommend a reading material for this person who is asking for the differences in the Cas?
Yeah, as I said, just read my last review that is being out now for a couple of months or for 1 month, and it's free. It's free access. So and if you wish, I could actually send you a PDF of it, and you guys distribute it, whatever you want.
Okay, we can if you can send us the link, perhaps later,
I can do.
Then we can provide them. Yes. So one of the questions that I have also is for Diane on what are the products that Two Blades have already been developed and already out there? And that's what I'm going to ask Jimmy later also.
And I'm guessing that, I'm not sure if that question means in general or specifically for gene editing. But the answer is the same actually in both cases, which is that we do not yet have products on the market, but we are developing resistance in a number of crops. I mentioned the wheat work. That was work that we've been involved with, and that's making progress. We also have advanced resistance in soybean against Asian soybean rust. We've worked in tomato, providing resistance to bacterial spot. We've worked in citrus. We've done work in potatoes against late blight. So we have a number of different programs, but these have not reached market yet.
Okay, but you see a lot of new improved crops through genome editing coming out very, very soon. So let's wait for those. And one of the questions for Jimmy is from Michael Jones. Is the sticker rice actually been grown commercially in China yet? Or what is the stage where it is at?
No,
now?
no, it is not commercial yet. And that was a proof of principle that could be done. By the way, we did it in 2 generations, but we did it only in a single year, because we used a bit of a trick. We actually grew one of the generations, we grew it in the northern part. And then the next generation, where we got the seeds, we put it on the southern part of China. So we could actually grow 2 generations in 1 year, but it's not commercial yet. It was a proof of principle.
Okay.
By the way, they're there and they're non-transgenic. So then again, you know, Professor Su is the one who's will take care of any kind of commercialization in China.
Yes. I am looking here at the questions, and most of them are on the differences between CRISPR and TALENs. And so generally, what can you say about maybe if you are advising newbies in genome editing, how would you suggest to them which one to use? What are the, is it based on crops or the traits that you're looking for? Or what are the factors that would help you determine to use TALENs or CRISPR?
There's both. They are both very effective platforms that can be used for gene editing. That's absolutely clear. There are probably individual cases where one may perform better or differently than another. I think as a guide, there's a couple of ways of thinking about it from the research perspective or commercial development perspective. From the commercial development perspective, there's intellectual property rights to consider, and having the appropriate licenses for either platform to take forward. From a research perspective, I think probably one of the influencing factors is going to be the crop. I'm not 100% up to date with which platforms are working best in which crops. There are probably some differences there. One of the distinguishing features that I mentioned about TALENs is that because it's just a protein editor, and that could be targeted inside an organelle, if you wanted to do organellar editing, TALEN might be the right choice for you. But I think, yeah, you would need to look at the literature and see what things have been successfully tried. Jimmy probably has more experience. I'm not currently in the lab doing these experiments directly.
TALEN, actually, TALEN was, when I went to China, I actually wrote my entire proposal on TALENs. And then, 2 months before going there, CRISPR was discovered and I had to change everything. So I never got to work with TALENs. Technically, there is no comparison. CRISPR is like 1 million times easier than TALEN in order to prepare the constructs that you need. Essentially, you just order 2 oligos and that's it. You order 2 oligonucleotides and that's it, and you have it cloned. As I say, you can clone. We clone 4 to 6 CRISPR constructs in an afternoon. So technically, for anyone who doesn't have a full proficiency in molecular biology, CRISPR would be the way to go because of the technical simplicity. Now, talent would be more precise because you use two different talents to target one single point in the chromosome. While CRISPR, you use one single sequence to do it. Having said that, we have done CRISPR experiments, at least at the beginning. And one time we got 20 different lines and we sequenced the entire genome. We didn't find a single off target. It's amazingly, good, in that respect. Then again, in plants, I understand that in humans it's important to be absolutely precise, but in plants, you don't really care if you introduce one mutation unless it's really a real bad one.
Yeah. Okay. Yes, you can use both actually. But I am really currently improvements in organelles is still new, or so that can be done through talents, and then the other ones can be done through CRISPR if you want quick and easy improvements, and quantitative traits just like yield can already be done through CRISPR. Because before I was thinking that it'll be impossible to do in genetic engineering. You cannot do quantitative traits, and yield is a quantitative trait, and it can be done through CRISPR if you can just design your targeting sequence that they will be able to target all those quantitative genes. Now, one of the things that also come out in the questions now is we're going now to regulation. We have tackled the science and the comparison. And one of the things here is the European view on genome editing. And we know that it is still being regarded by Europe as genetic engineering. So how do you think you will approach the regulation of Europe? How can we explain to them that this is just mutation, this is in comparison to genetic engineering
we don't need to explain them anything, they know.
They know, okay.
Yeah, what Europe is doing is a political thing. It's got nothing to do with science. It got nothing to do with reality. It has to do with a number of groups that have important lobbying into the European Parliament, okay? And those groups are normally, influenced by philosophies rather than scientific facts. So in Europe, they know. I'm European. I grew up in Europe. They know what is the reality. The thing, whether they approve it or not, is a different thing. It got nothing to do with reality.
Yeah. Yes. Can you say something about that? The question here is for Diana, actually, and the question is from Max Planck Institute in Potsdam. So it's a German.
Yes, I saw the question from Augustine. I can't really comment on European trade policy to say anything meaningful there. But certainly Europe does influence a lot of what happens in other parts of the world, especially in Africa and crops. I think my concern more for Africa and Asia is really the ability to have control over agriculture to become more efficient, both for the nutritional aspects, but for the economic impacts. We know that strong agricultural economies help lift up people and not only their health, but also their economic well-being. So I really hope that these tools can help to benefit smallholder farmers in developing agricultures, first and foremost for them.
Yeah, and actually we have already discussed also that the precision in the non-regulation of genome edited products will make it more available to needy people, the developing countries who need the food. And this will also expedite the improvement of different types of crops, different types of traits. And so we can say that the talents and CRISPR are both important tools in innovating and improving our crops, not only the crops, but also in animals and other microorganisms. So with that, would you like to say your last few words of some science or some key takeaways on your innovative tools, please? Let's have Diana first.
So one follow-up to the point that Jimmy made a moment ago. Certainly, the CRISPR system is so easy and elegant to use. It has a lot of advantages, especially for researchers, just to be able to quickly and cost-effectively test things in the lab. But I do want to point out that the new assembly systems for TALENs make it really also very fast. There's now 1-hour assembly systems. So I think there's still plenty of value. And although multiplexing also is not as simple in TALENs, it is possible. And I think both are evolving all the time. So I think both are going to be important tools. Gene editing is important. Transgenic is important. I think we all want to be very clear about our messaging, not only for policy and making sure these products can reach benefits for people, but also to consumers to make sure that they understand the safety and importance of these tools. So I hope that we can see continued growth and use of these tools and the benefits of agriculture, because there are plenty of challenges to produce adequate food in the right places for everybody.
Yes, definitely. Thank you so much, Diana. Jimmy?
I totally agree with Diana. It's we need a lot of tools. We are in a lot of trouble. All right. Climate change is coming. We're having more and more extreme events. Human population keeps going through the roof. We are ruining our ecosystems. We need all the help we can get. Genome editing, all kind of technologies we need to be combined in order to feed humanity and save the environment, because the way we're going now with agriculture is not really sustainable.
Mm-hmm. Yeah. And one last question which came up is, which is better to use, genome editing or gene editing? This is from Michael Jones.
Well, it's pretty much the same. There are synonyms on there. But let me tell you, just to finish, one thing that is really interesting that we have done. You can actually affect a gene that have two different traits. We have genes, for example, that are very important for defense and are also important for meristem development. What we've done with genome editing is to target different parts of the promoter. And there are parts of the promoter that are really specific for defense and other parts that are really specific for meristem development. We have been able to alter the meristem traits without affecting defense on the single gene. So forget about the genes. There are a complete new set of applications looking away from the genes in the, in the promoter regions, that you can actually end up doing things that are almost impossible to think about, and those are coming. We have done it and it works really well.
So it's genome editing, not specific gene editing.
Yes,
because you
exactly.
can target anywhere. Okay, thank you. Yes. So currently we are now, do you have any other questions? Okay, or any additions that you can say before we close, before we give the floor to Margaret? Our dear Dan. But yeah,
yes, thank you for this opportunity to debate these important topics and try to inform and educate people about science and technology and improvements and innovations. It's great to have the opportunity. And thanks to ISAAA for hosting these discussions.
Thank you so much.
Yes, I want to thank ISAAA too. I mean, you guys are doing a fantastic job. I would like to offer myself, if there are teams that want to start thinking about genome editing and they need some advice, just please contact me. I'll be happy to provide advice.
Okay, thank you so much. Let's give them a round of applause, virtual. So we now give the floor to Margaret.
Thank you so much, our speakers and our participants. There are still questions, but some of them will respond, one-on-one. I saw one from Kenya asking about use of CRISPR to introduce productivity in rice. And I was just wondering whether, between Iri or even Jimmy or Diana, has there been, are you using any of these tools for rice? I know the sticky rice.
Yes, it has been done. There are plenty of work now proving that there are high improvements in yields, improving in nitrogen use efficiency, meaning that they need less fertilizer. So it has been done in rice, yes.
Okay, so basically, of course,
Diana will say something also.
Diana, yeah.
I know Iri is very active in gene editing. I don't know the full extent of the traits that they're looking at. I believe that they're looking at blight resistance, biofortification, a number of traits, but I'm not up to date on all of them.
And TALENs has a partnership with Iri on this.
That's right.
Improvements.
Okay. Thank you so very much. I think these are opportunities, especially even the offer for our young scholars in our universities. This is now an opportunity to really bring products to the market quickly, and we do this seminar so that we can also create these linkages. So if you're out there, a young career professional in biotechnology, and you'd like us to link you more with Jimmy. Jimmy has already offered that he's willing and ready. Diane and her team at Two Blades is also willing and ready to work with us. And I think the question of Europe is a very sticky one, because the position they take affects all of us in one way or the other. Either the developers who would like to help to work with other countries to bridge this big gap between food insecurity, improvement in the environment, and yet, it is very difficult also to challenge them, because they'll tell you, do as you wish. It cannot be free for all in a scientific world where people are sharing resources, sharing knowledge. So we really hope that this time round, the solution will be just to get the African and the developing country scientists to move quickly, not to really follow what others, to move quickly, demonstrate to our policymakers that these tools are useful, they can address many of the challenges we have, and hopefully we'll not have to just rely on ideologies. So thank you so very much. This was very, very useful, and I hope the conversations will continue. So now we are getting to almost the last part of our webinar, where I'll invite our global coordinator, Dr. Mahalakshmi Alujanan. We just call her Maha.
Yes, just Maha.
To do the closing remarks as she shares the new direction for ISAAA. So thank you very much. And remember, our participants, don't go away, because we still have our evaluation survey.
Actually, it's already posted at the, excuse me, the ISAAA website evaluation form is already posted at our chat. So attendees, please look at the link, and this will be the basis of your certificates. Thank you. Okay, Maha.
Thank you so much, Margaret. Thank you, Ola, and thank you, Diana and Jimmy, for all your support. And we are so happy to be hosting this, because we have already moved into ISAAA phase 2, which I'm going to talk about. I won't take so much time, but let me share my slides. Okay, so ISAAA has been in this space for a very long time. So we have been a pioneer in knowledge transfer and agri-biotechnology for almost 3 decades now. And we have done so much of impactful work in Asia and Africa, these 2 regions. And what we have done is we have facilitated approval and adoption of biotech crops. But today you see us moving into gene editing and other technologies, which I will share with you shortly. We have supported resource-poor farmers and alleviation of poverty, increased public awareness on agri-biotechnology among all stakeholders, and also capacity building on biosafety and also communication. The uniqueness about ISAAA is ISAAA has been built as a reputable, non-partisan, internationally accepted, credible source for information. And as I said, with 2 strong regional offices. One is in Southeast Asia. Ola is a director, and you've seen her work. And the other one is Margaret in Africa. But we are also expanding to Arab and Latin America, and we are looking at working very closely with stakeholders in Europe as well. So I really feel this is very exciting time for ISAAA. Now, we also know the concern at the background is while science is moving very fast, rapidly advancing very fast, we still see so much of opposition, so much of concern. So it is like a post-truth, post-trust, and post-expert era. And COVID vaccines and COVID is showing us that this is true. As much as we are also advanced, there's still so much of opposition, hesitancy towards science. And this is where we need to really work harder and bring in new strategies. So this rapid development, but then when we compare to public understanding, there's so much more work to be done. So with that background, ISAAA is now moving towards the next phase, and we call ourselves Biotrust Consortium. So as a consortium, we are inviting partners, like-minded partners. Two Blades is a perfect example to work with us to achieve our common goal. Our tagline is to be a global leader in building trust and transparency in biosciences. If you note the word, it is biosciences. It is not just agriculture. Agriculture will be the root, but then from there, we have so many other new technologies coming in. So we want to remove ignorance and fear. We want to build, trust and transparency. We want to obtain social licensing. We spoke about Europe. Now, we know these are all approved by regulators, but approval and adoption are two different things. So now we want to build the social licensing, the license, the approval that comes from the public and community. So we need to have that. So we need to really engage the public and all the other stakeholders besides farmers. And that is what we're looking at now. Our purpose statement is to, let me move this, is to improve society's acceptance of novel food and new food systems through multi-stakeholder trust building activities. So we have been working very closely with farmers. Yes, we have been working with all the other stakeholders, but now we want to really move to working with many other players in different industries in biosciences. Our vision is a world where science contributes every day to prosperity, social well-being, and sustainability. And I always use SDG goals, SDGs as an example. I feel most of the SDGs can be achieved if we really embrace science. So we are going beyond crop biotechnology for sustainable food production and environment. We are widening our scope to support sustainable food production and socio-economic growth. So we are looking at new breeding technologies, just like what we're doing today. And even last year, we started that. And we did a webinar series on gene editing. And it was so successful. We had 97 countries participating with 60,000 participants. So we are looking at biomass. How can biomass be used as green technology to convert biomass into green products? We are moving into from crops. We are also looking at livestock, poultry, aquaculture. In fact, last year, we also did a series of webinar on animal biotechnology. We're looking at microbial technologies or precision fermentation. I think this is really an exciting field, the industrial biotechnology field, but still with a root in agriculture. Future food, lab made meat and plant-based meat, and also science literacy and social licensing. So how do we, what will Biotrust do to build trust and transparency? We're looking at inclusive engagement strategies where we want to reach out to all stakeholders in food systems, all communities in all countries. We are looking at also facilitating social licensing, as I mentioned before, reaching out to the grassroots, where if we see in Europe, the general public are the one who is shaping the public policy opinions and the opinions of policymakers. So we want to reach out to those people. Capacity building for developing countries. We want to do more capacity building for developing countries. And this is to support social justice on access to technology. So this technology should reach out to every country, every region. We want to ensure transparent knowledge sharing. So being a non-partisan knowledge sharing, being transparent about all this technology, and also future-proofing society on novel foods and biosciences, because this is definitely going to change the landscape of how food is produced. And what builds the foundation for Biotrust Consortium is our past, the history. ISAAA has got probably the highest followers and subscribers among all those like-minded organizations that are in this space. We have got, the latest is about 193 actually K followers in all our platforms in social media. As I said, we are very active in 2 regional centers, and we also have 15 country chapters in Southeast Asia and Africa. We've got the experience, network and expertise of about 30 years being in this space. We have built our credibility among global stakeholders in this space. And coming up is Arab, Latin America, and Europe. And we are also getting very active in Australia. So, you know, it's really an exciting time for ISAAA, and we hope more partners will join us. So we are building this consortium, hoping that, you know, to open it up for membership. We will fine-tune this further, but we're looking at how we can have core members, where, you know, members providing annual funding to run the secretariat and technical team, how they can also have a role in deciding programs that we run. And we can have supporting members as well, with some sort of in-kind support to support our operational cost. And also knowledge partners, where we share our strength and we share our resources to achieve our common goal. So this is what we're looking at. Of course, we're still fine-tuning our organization to move forward, and we invite everyone to join us and also to provide input. So please speak to us so that we can achieve our common goals. And thank you very much.
Thank you so much, Maha. So our speakers, our staff and participants, we have come to the end of our webinar. It has been an exciting 1.5 hours. We'll definitely continue with networking. Please contact us. There's still conversations that are going on the chat, and we'd like to continue with those maybe on one-on-one. And we'll continue with this discussion, and hopefully we'll also make stronger linkages with the 2 blades already with the coming on board, and also University of Queensland. With the 2 companies that Jimmy has formed, we look forward to having them in our consortium. So thank you so much.